In continuation to the previous session, we are going to see in this session heteropolysaccharides. So heteropolysaccharides are similar to that of your homopolysaccharides. The major difference is the monosaccharide unit which is present in your heteropolysaccharide will be different. So 2-3 monosaccharide units can combine to form heteropolysaccharides. So in case of homopolysaccharide, we saw it is made of same monosaccharide units. Whereas in case of your heteropolysaccharide, you will have different monosaccharide units, again, that are linked with your glycosidic linkage. And usually, this kind of heteropolysaccharide will be used for extracellular support. And also you can see this kind of heteropolysaccharide in case of your bacterial cell envelope, especially the peptidoglycan layer, which is present in the bacteria cell wall. So this will have heteropolysaccharide units. And here for this heteropolysaccharide also, you can't define the molecular weight because the monosaccharide units can exist from 20 to 100 or sometimes 1000 or above 1000 monosaccharide units which can be present. And one major important function of this homopolysaccharide and heteropolysaccharide, irrespective of whether it is structured or storage component, these are water insoluble in nature, whereas whatever we have studied previously, the monosaccharide are soluble, sucrose will be soluble in water, whereas in case of your polysaccharide especially, whether it is homopolysaccharide or heteropolysaccharide, these are not soluble in water. That is why in the previous class, we saw homopolysaccharide, which includes cellulose and chitin. So these are all protective agents. So you can see that in cell wall, animal exoskeleton, so that it prevents water, so that it will not allow any of the water molecule which can go inside, and it will avoid osmosis process. So both will be insoluble in water. And this polysaccharide, you can just do a simple test. If you just take the polysaccharide, add 1 drop of iodine, you will be getting blue color. So the presence of polysaccharide is identified with the help of iodine. So iodine test you can do, a drop of iodine, if it is added to the polysaccharide, you will get blue color. So if you get blue color, it is the presence of polysaccharide. Whereas if we perform the same iodine test with glucose or with monosaccharide or with disaccharide, you will not get the blue color. So if blue color is present, then confirmation, the test says that polysaccharides are present, whether it is homopolysaccharide or heteropolysaccharide, polysaccharide is present in the given sample. Now again, like homopolysaccharide, this heteropolysaccharide is also classified into 2 types. One is the linear structure, the other one is branch structure. So this classification remains same as that of the homopolysaccharide. So here also you have linear heteropolysaccharide and branched heteropolysaccharide. You can see each one are in different colors, which denotes you have different monosaccharide units. This may be glucose, the other one may be galactose, the green one may be mannose. So like that, you will have different monosaccharide units which will be made up of, and you will be getting the heteropolysaccharide. So if it is unbranched or linear, you will have same glycosidic linkage with alpha 1,4 most of the times. And if it is branched, in the branch point, again, you will have alpha 1,6 linkage. The remaining linear structures will be having alpha 1,4 linkage itself. So your polysaccharides are classified into homopolysaccharide as well as heteropolysaccharide. Under heteropolysaccharide, again, we have linear and branch. Under homopolysaccharide also, we have linear and branch. So both we have linear and as well as branched structures. So we'll see some of the examples of heteropolysaccharides. So heteropolysaccharide, which includes hyaluronic acid, this hyaluronic acid will be present in the connective tissue between the joints and all. You will be able to see this kind of hyaluronic acid. And also in the skin, you will have hyaluronic acid. So the major function of hyaluronic acid is it is acting as lubricant, shock absorber, water binding and all. And if you see the components of the monosaccharide, you will have D-glucuronic acid and N-acetyl glucosamine. So glucosamine is one of the monosaccharide unit, and Glucuronic acid is also one of the monosaccharide units. So these are the two different monosaccharide units which will be present in hyaluronic acid. And the next example is chondroitin 4-sulfate. And in the chondroitin 4-sulfate, we have D-glucuronic acid and N-acetyl galactosamine 4-O-sulfate. So these are the two monosaccharides units which are present. And of course, it will be present in cartilage, and it is used for calcium accumulation in cartilage as well as in the bone formation. And the third heteropolysaccharide is heparin, and heparin is composed of D-glucuronic acid, L-ioduronic acid, and L-sulfo-D-glucosamine. So these are the three monosaccharide units which will be present in the structure of heparin. Of course, it will be present in blood as well as in the mast cell, and this will be used as anticoagulant. So this heparin is present in the blood in order to avoid the coagulation when it is inside the blood vessel. The fourth one is gamma globulin. Gamma globulin is made up of N-acetyl hexosamine, mannose, and galactose. So three different monosaccharide units which will be present. And of course, this will be seen in blood. And of course, this is used as antibody. The last one is blood glucose substances. And these antigen antibody substances are made of D-glucosamine, D-galactosamine, L-fucose, and galactose. These are the four different monosaccharide units which are present in blood group substances. And this you can see in the cell surface, especially in the RBC cell surface, you will be able to see the blood group substances. And the major function is to identify different blood groups. So blood group specificity will be determined with the help of these carbohydrates which are present in the blood group substances. So you have hyaluronic acid, chondroitin 4-sulfate, heparin, gamma globulin, and blood group substances. So these are the heteropolysaccharide with different functions in your body. It is located in different places of your body with the different monosaccharide units which are present. With this, we complete the carbohydrate unit. We'll see the other units in the upcoming sessions. Thank you, students.